Dear students, today our topic is regarding morphology, anatomy and reproduction of Selaginella. Selaginella, with about 700 species, is cosmopolitan in distribution. It is commonly known as spike moss or small club moss. Most of the species inhabit damp and shaded forests of tropics, but some grow in xerophytic habitats, such as exposed rock surfaces. Selaginella oregana is an epiphyte that grows on tree trunks in tropical rainforests. Several species of Selaginella are grown in gardens as ornamentals. Some xerophytic species of Selaginella, that is Selaginella lepidophylla, Selaginella pilifera, show cespitous habit. They curl and become ball-like during dry season and again become green and fresh when moisture is available. These are called resurrection plants. The genus is represented in India by more than 70 species. Among these species, Selaginella cruciani, Selaginella monosporma, Selaginella biformis and Selaginella rupestis are common. Now we will know about its morphology. Morphology. The sporophyte of Selaginella is differentiated into root, stem and leaves. Besides, some species also have rhizophores. Roots. Primary roots are epiphyral and adult plants have adventitious roots. The adventitious roots usually have specific locations in relation to stem dichotomies. In most of the creeping species with dorsiventral stems, for example Selaginella cruciani, Selaginella levigata, roots arise at or close to the point of dichotomy. In species like Selaginella rupestis and Selaginella walshi, they arise at the point of dichotomy as well as at the other positions. And in Selaginella seliginoides and Selaginella spinulosa, they arise from knot-like swellings present at the basal portion of the stems. The roots arise endogenously and are dichotomously branched. The dichotomies are at right angles to each other. Rhizophore. In some species of Selaginella, many long, cylindrical, unbranched, leafless structures arise from the lower side of the stem at the point of dichotomy. These grow vertically downwards and bear tough adventitious roots at their distal end. They are known as rhizophores. Now stem. The stem is erect and dichotomously branched in the subgenus Homophyllum and prostrate or sub-erect with lateral branchings in the subgenus Heterophyllum. The stem apex usually has a single well-defined apical cell, but in Selaginella oregano, a group of meristematic cells has been observed. Now leaves. The leaves of Selaginella are microphyllous and simple. Their shape varies from ovate to lanceolate. The leaf has a single mid-vein that remains unbranched throughout its course. Most of the species have thin and soft leaves, but in xerophytic species they are thick. The vegetative leaf as well as sporophyll has a new membranous projection on its adaxial surface close to the base. The projection is known as ligule. The basal part of the ligule has a distinct hemispherical foot-like structure called glospodium. It is composed of highly vacuolated thin-walled tubular cells. Although the definite function of the ligule is not known, it has been suggested that in some way they are associated with water absorption and secretion, thus prevent desiccation of the shoot. Some consider that ligule in Selaginella are concerned with upward movement of inorganic solutes and thus compensate for smaller and less effective leaf primordia. In the subgenus Homophyllum, all leaves are alike and spirally arranged, but the species belonging to the subgenus Heterophyllum have two types of leaves, two dorsal row of small leaves and two ventral row of large leaves. The leaves occur in pairs and the two leaves of a pair are always unequal. Now anatomy. Root. First of all, root. A cross section of the root shows a simple structure. The epidermis is made up of tangentially elongated cells. In exposed roots, the outer wall of the epidermal cells is cutanized, but in roots that penetrate the substratum, the epidermal cells are delicate and have root hairs. The cortex is usually homogeneous, consisting of many layers of thin parenchymatous cells. But in some species, the outer layers of the cortex became thick, walled, and form hypodermis. The innermost layer of the cortex forms endodermis. The central part of the root is occupied by a protostele, surrounded by 1 to 3 layers of parenchymatous pericycle. The xylem, which forms the central solid core of stele, is monoarch to tetrarch and exarch. The phloem occurs in the form of a ring around the xylem. Rhizophore. The structure of the rhizophore resembles the root. Some variations in the internal organization are due to the fact that rhizophore is an aerial structure, whereas the root is a subterranean organ. The epidermis is single-layered and the outer wall of the epidermal cells is covered with a thick layer of cuticle. Root hairs. Characteristics of roots are absent on rhizophores. The cortex is differentiated into an outer sclerenchymatous and an inner relatively wide parenchymatous zone. The innermost layer of the cortex forms endodermis. The protostele of the rhizophore is surrounded by parenchymatous pericycle. Usually the stele is monoarch and exarch. Stem. The anatomy of the stem shows variations not only in different species but also within the same species depending on the stem diameter. A transverse section of the stem shows epidermis, cortex, and central cylinder. The epidermis is the outermost unistratous layer. The outer walls of the epidermal cells are highly cutanized. The epidermis is devoid of stomata and hairs. The cortex is usually composed of compactly arranged parenchymatous cells without intercellular spaces. But in mature stems of many species, outer layers of the cortex becomes partially sclerenchymatous, forming a tough hypodermis. In xerophytic species like Selaginella rupestris, Selaginella lepidophylla, most part of the cortex is composed of thick-walled cells. A distinctive feature of the Selaginella stem is the presence of radially elongated endodermal cells called trabeculae. They have a characteristic Casparian bands on their lateral walls. Due to the presence of trabeculae, the central stele is separated from the cortex by large air spaces. The air spaces develop due to more rapid expansion of the cortical region than the stele. The number of steles in the stem shows considerable variation in different species of Selaginella. For example, the stem is monostelic in Selaginella spinulosa and Selaginella flabellata, distelic in Selaginella cressoni, and polystelic with 12 to 16 steles in Selaginella levigata. Besides, the number of steles may also vary with different parts of the same plant. The stele is surrounded by a single layered pericycle. It is circular in Selaginella cruciani and flat or ribbon-like in Selaginella verandicula and Selaginella vigili. Most of the species have protostele with a solid core surrounded by phloem. The xylem is usually monarch, that is Selaginella cruciani, or diarch, that is Selaginella seliginoides. It usually consists of only tracheids. The protophlloem tracheids have annular or helical thickenings, whereas the metaxylem tracheids show scalariform thickenings. In Selaginella origana, Selaginella densa, and Selaginella rupestris, however, the xylem has two vessels with transverse perforation plates. Now leaf. Both upper and the lower epidermis of the leaf are unistratous. The epidermal cells have chloroplast. The leaves are mostly epistomatic, but sometimes they are hypostomatic, as in Selaginella martense. Stomata are distributed mostly in the midrib region. The mesophyll consists of loosely arranged thin-walled cells with many small or large intercellular spaces. It is usually made up of only spongy parenchyma. A mesophyll has 1 to 8 cup-shaped chloroplast which has many spindle-shaped pyramid-like bodies. The leaf has a median vascular bundle surrounded by distinct bundle sheath. The xylem, which occupies the central part of the bundle, consists of only trachearis with annular or spiral thickenings. It is surrounded by phloem. Now, reproductive part of Selaginella. Selaginella reproduces vegetatively and by spores. Now, vegetative reproduction. Vegetative reproduction in Selaginella takes place by tubers, bulbils, dormant buds, and by fragmentation. In Selaginella rupestris, prostrate branches produce roots during favorable conditions. These root-bearing prostrate branches separate from the parent plant and grow into a new sporophytes. Species like Selaginella chrysorhizos and Selaginella chrysocollus propagate with the help of tubers and bulbils. The tubers may be aerial, developing at the apices of aerial branches, for example, Selaginella chrysocollus, or subterranean, for example, Selaginella chrysorhizos. During favorable conditions, the tuber germinates into a new sporophyte. Aerial branches of the Selaginella chrysocollus also bear some dormant buds which grow into new plants during favorable conditions. Reproduction by spores. Selaginella is a heterosporous pteridophyte. It produces two types of spores, megaspore and microspores. The megaspores form female gametophyte on germination, and the microspores give rise to male gametophytes. The sporangia are strictly dimorphic, that is, micro and megaspores are formed in separate sporangia. The sporangia are born singly in the axils of the sporophylls. The sporophyll bearing microsporangium is called as microsporophyll, and the one with megasporangium is known as megasporophyll. The sporophylls are spirally arranged around a central axis. Strobilus or cone. In most of the species of the Selaginella, sporophylls are aggregated at the apex of the main stem and its branches in a definite loose or compact cones called strobili. The size of the strobilus varies from 5 mm to 7 mm. It is often inconspicuous due to its small size and similarity between sporophylls and vegetative leaves. Now, development of sporangium. The initial stages of development of micro and megasporangium are similar. Both develop from the transverse row of initial cells, that is, development is of eusporangiate type. The sporangial initials divide spirally, establishing outer jacket initials and inner archisporial initials. The archisporial initials undergo repeated anticlinal and periclinal divisions, forming a mass of sporogenous cells. Simultaneous divisions also occur in the jacket initials, and the derivatives eventually form a two-layered sporangial jacket. The cells of the outermost layer of the sporogenous tissue adjacent to the inner wall layer form nutritive layer known as tapetum. The tapetal layer disintegrates as spores mature. The last generation of the sporogenous cells function as spore mother cells. The micro and megasporangium differ in subsequent development. Now, mature sporangium. Mature sporangia are stalked structures with two-layered sporangial jacket. The cells of the outer jacket layer are elongated and contain chloroplast. The micro and the megasporangia differ in size, shape, and color. The microsporangia are slightly elongated, yellow, red, or orange in color. The megasporangia are larger and paler and assume the shape dictated by the enlarging megaspores within. The mature megasporangium dashes along the line of dehiscence present at its distal end and oriented transverse to the axis of the sporophyll. Structural modification of the surface cells along this line and at its flanks results in the splitting of the distal part of the sporangium into two valves. The lower cup-shaped part of the sporangium shrinks on drying and throws out spores violently. Now, gametophyte. The spore is the mother cell of the gametophytic generation. As mentioned earlier, Selaginella is heterosporous and produces two types of spores, the smaller microspores and the larger megaspores. The difference in the size of spores is related to their fate and function. Microspores develop into male gametophyte and megaspores into female gametophytes. In Selaginella, both micro and megaspores begin to germinate while still inside the sporangium, that is, they germinate in situ. Thus, spores are shed at multicellular stage. Now, development of male gametophyte. The microspores germinate inside the microsporangium and are shed at 13-celled stage. The first division of the microspore is asymmetrical, and as a result a small lenticular prothelial cell and a large enthedial initial is established. The prothelial cell does not divide further and the entire sporangium develops from the enthedial initial. The first division of the enthedial initial is nearly at right angles to the prothelial cell. It results in the formation of two enthedial cells of almost equal size. Both these cells divide by a vertical wall to produce a group of 4 cells. Thus, at this stage, the gametophyte consists of 5 cells. That is, 4 cells derived from the enthedial initial and a prothedial cell. The 2 basal cells derived from the enthedial initial do not divide further, whereas the upper 2 daughter cells divide repeatedly and form 10 cells. At this stage, the gametophyte has 13 cells. That is, 10 cells derived from the upper daughter cell of the enthedial initial, 2 basal daughter cells and 1 prothelial cell. Of these, 4 central cells function as primary androgonial cells and 8 peripheral ones as a jacket cells. That is, male gametophyte is shed from the microsporangium at 13-celled stage. It enters into a partially opened megasporangium where further development of the male gametophyte takes place in close proximity of the developing female gametophyte. In some species, it is believed that further development of the male gametophyte takes place in the soil. The 4 central primary androgonial cells of the male gametophyte divide repeatedly, forming a mass of 128 to 256 anthrozoid mother cells or androcytes. Each androcyte metamorphizes into a spindle-shaped biflagellate anthrozoid. The anthrozoids of Selaginella are perhaps the smallest amongst the vascular plants. With the formation of anthrozoids, the jacket cells decompose and form a mucilaginous substance. The anthrozoids float in this substance. Until this stage, the male gametophyte is completely enclosed within the wall of the microspore. Thus, it is entirely endosporic and extremely reduced structure. Unlike other pteridophytes, vegetative prothelae are not formed in Selaginella. The gametophyte is not set free and is dependent on the parent sporophyte for nutrition. Now, megaspore and the development of female gametophyte. Megaspores are much larger than microspores. The diameter varies from 0.15 to 0.5 mm. Usually, all the megaspores in the megasporangium are approximately of the same size and are arranged in tetrahedral tetrads. Now, the development of female gametophyte. Like male gametophyte, the development of the female gametophyte of Selaginella also begins while it is still within the megasporangium. Immediately after the development of female gametophyte, a large vacuole appears in the center of the megaspore, and as a result, the cytoplasm is pushed along the spore wall in the form of thin membrane. There is a considerable enlargement of the megaspore. The outer spore wall exposes, that is, exospore grows more rapidly than the mesospore and endospore. Consequently, a large gap is formed in between the exospore and mesospore. At this stage, the exospore is attached to the mesospore only at one point. The space between the exospore and the mesospore is filled with homogeneous liquid. The haploid nucleus of the megaspore divides repeatedly without any wall formation. The free nuclei are unequally distributed in the peripheral cytoplasm. They are clustered beneath the triradiate ridge of the spore and sparsely distributed elsewhere. Now, Wall formation begins in the apical region and a lens-shaped pad of a small cells is formed at the apical end. It is separated from the rest of the female gametophyte by a distinct diaphragm. The cytoplasmic layer becomes thicker gradually and pushes the mesospore outward. As a result, the mesospore again comes in contact with the exospores. With the increase in the amount of cytoplasm, the central vacuole diminishes and eventually disappears. The part of the gametophyte below the diaphragm is multinucleate in early stages but becomes multicellular as wall formation proceeds inward. At this stage, the spore wall ruptures along with the triradiate ridge, exposing the apical cellular pad. The exposed part of the female gametophyte may develop chloroplast, but the photosynthetic ability of this part is of limited importance as the food for the developing embryo is stored in the lower multicellular part of the gametophyte. Many rhizoids develop from the exposed part of the gametophyte. They attach the gametophyte to the substratum and also help in absorption. Now, development of Archegoniae. Archegoniae develop from the apical tissue of the gametophyte. All superficial cells of this tissue have potential of forming Archegoniae. The Archegonial initially divides pericardially into primary cover cell and a central cell. The primary cover cell divides by two vertical divisions at right angles to each other and forms 4 neck initials. The neck initials divides transversely so as to form 8 neck cells arranged in 2 tiers of 4 each. In the meantime, the central cell divides by a periclinal wall and an outer primary neck cell and an inner primary ventral cell is established. The former does not divide further and directly functions as neck canal cell, whereas the latter divides transversely into a ventral canal cell and an egg cell. Now, fertilization. Fertilization usually takes place after the megasporangium has fallen on the soil, but in some species it may occur while the female gametophyte is settled within the sporogenium. Just before fertilization, the neck canal cells of the Archegonium separate from each other and form a passage for the entry of enterozoids. After liberation from the male gametophyte, the enterozoids swim in rain or dew water to reach the Archegoniae. Usually only one enterozoid enters into the Archegonium and fuses with the egg to form a diploid zygote. Some species of Selaginella like Selaginella rupestre, Selaginella apoda show seed habit. In these species, the sporangium has only a single megaspore and at maturity of the Archegonium, the spore wall ruptures. But the developing female gametophyte does not come out of the spore wall. Male gametophyte, when shed from the microsporangium present in the distal part of the strobilus, lands on the partially open megasporangium. Thus, at this stage, both male and the female gametophytes lie within the megasporangium. As such, fertilization and embryo development takes place inside the megasporangium. The sporangium is shed after the development of root and a primary shoot of the new sporophyte. This feature is of a considerable importance from the point of the view of the seed habit because when the megaspore with young sporophyte is shed, it has all typical characters of seed. In early stages of development, the young sporophyte is attached to the megaspore and derives its food from the female gametophyte with the help of its foot. But after the establishment of the root and the stem, the sporophyte becomes independent. Dear students, with this we conclude our today's topic, that is morphology, anatomy and reproduction of Selaginella. Hope that you have got it. Thank you once more.